offering a new class this summer. It's experience in plant sciences, extension and outreach. And it's cross-listed with agronomy and horticulture and entomology. And I just wanted to draw your attention to it because you can now sign up and register for it. I know that a lot of you, when you think about summer, you think about, yay, this is a time where I can really knuckle down and I do my research. And I know a lot of your advisors want you to knuckle down and do your research. And so I wanted to just let you know that one thing about this class is it really doesn't take a lot of time. We're going to be having one class period approximately every 3 weeks, and that'll just be for an hour and a half, where we'll actually meet with a class and we'll be with students from entomology, agronomy, and horticulture as well. So we hope to get very diverse people. And then the rest of the time, the classes, you'll be working by yourself or through Canvas, and with a faculty advisor of your choosing. So from this department, there'll be Darren and I and Laura and Lena. And then outside of the departments, if you know them, Mark Licht, RJ Nair, and Erin Hodgson. So I just wanted to, if you have any questions, please feel free to come and talk to Darren or I, Lena or Laura in the clinic. And we really encourage you to sign up. So it's PLP 581X.
And then, the other thing is that it will fulfill a requirement that we have for your MS or PhD for a communications course. So there's another benefit there.
So if you're interested in extension, you want to see what it's all about, this is just just an idea, just to give you an idea of what we do in extension.
Okay.
Thanks, Gary.
All right. Well, in my job with responsibilities in seed pathology, it often steers me into territory where I don't know what I'm doing, and so when that happens, I try to find somebody who knows a lot more than I do, and that's kind of what ended up bringing Tony here with us today. We're doing some work on some of the cucurbit diseases, like stem blight, that Tony's an expert in. And so glad to have him here to talk to us about his work on that disease. Tony has a PhD from Cornell, where he worked on potato scab. You can tell me when that was exactly, but maybe we're not talking about that today. It's about the same time as me, I suppose. And then after that, he did a postdoc with USDA in Beltsville, working on verticillium wilt in potatoes. And then in 1991, he went to Clemson to start his position as a vegetable pathologist. So now he's a professor in the department there, and he has a 80% research, 20% extension appointment in vegetable pathology. So with that, I will turn it over to Tony, and thanks for being here.
Right. Thanks, Gary. I'd like to thank both Gary and Mark for the invitation to come and talk to you about my favorite pathogen this afternoon, the gummy stem blight fungi. I have to say gummy stem blight fungi now, because a major point of this seminar is to introduce you to the 2 new species that cause gummy stem blight on cucurbits. Even though my appointment is only 20% extension, I've given a lot more extension presentations than research presentations during my career. So I would like to invite you to ask questions during my presentation, if you wish, instead of holding them to the end like in a traditional seminar. But before I get started, there's a couple interesting similarities between Clemson and Iowa State that I'd like to mention. Well, first, one unique thing about your department is that I think you were the only department in the country to have 2 Clemson graduates on your faculty, and that, of course, are Thomas and Allison. My current master's student is an Iowa State graduate. He got his undergraduate major was in agronomy, but he got his start in plant pathology in Allison's lab. But the third thing is not quite so cheerful, and that is the recent flooding here in Iowa. We certainly heard a lot about that in the news. And growers in South Carolina have also had to deal with significant flooding mainly from hurricanes in 2015, 2016, and 2018. But we certainly hope that either the state or the federal government provides some relief to your growers, as the state government did in South Carolina. Okay, so first we'll start with a short introduction to gummy stem blight. Then we'll talk about the taxonomy and nomenclature that changed one causal agent into 3 causal agents. And then on to the major topics this afternoon is how these species spread. They spread on land, in air, and I'm going to propose that they can also spread by sea. So the disease triangle, of course, starts with a nice healthy crop of watermelon or other cucurbits, and the pathogen, Stagonosporopsis, shown here as pygnidia. They have typical phoma, pygnidia, globose, tan when they're immature, or mature but not necessarily dried out, and with a dark ring of cells around the oscule. And as someone who's mainly interested in the pathogen, I probably spend a little too much emphasis thinking about the pathogen and not thinking enough about the environment, which is absolutely critical for the epidemiology of gummy stem blight. And as you might guess from that rain cloud, the fungus is heavily dependent on moisture, and that is leaf wetness periods. So, for many pathogens, you put them into a moist chamber after you inoculate during the infection period. Well, for gummy stem blight, you cannot take those plants out of that moist chamber and put them on the greenhouse bench, because you will hardly get any disease development at all. You have to leave them in continuous moisture for 3 to 4 days before you get significant disease development. So the fungus requires moisture not just for spore germination and infection, but also for lesion expansion. And I think it's kind of unique in this respect, certainly among the other foliar pathogens on cucurbits. When I started working on downy mildew and anthracnose, I was quite surprised that they didn't need as much leaf wetness as Stagonosporopsis does. And so when those 3 conditions come together, you have typical disease development. Shown here is these dark lesions with target-shaped appearance inside of them on watermelon. The target shape does not necessarily develop under all conditions or under all hosts, but the large lesions that rapidly expand under moist conditions are very typical. And here you see a typical disease cycle for gummy stem blight. Of course, seed here can be a primary source of the pathogen, but I think since watermelon seeds are being tested for bacterial fruit blotch by grow-outs, where 10,000 seeds per seed lot are planted in a greenhouse and grown for 3 weeks under warm, moist conditions for development of symptoms of fruit blotch. Symptoms of gummy stem blight will also develop under these conditions. And so the seed companies can remove seed lots that are infested with the gummy stem blight fungi from the market. And I would say that the incidence of gummy stem blight on transplants has decreased in the last 10 years. But you can get symptoms on seedlings, particularly on muskmelon. You can get severe stem cankers. You can have high percentages of the plants in the field affected with stem cankers that don't normally kill the plant, but they are an important source of inoculum for the foliar blighting. Gummy stem blight is also one of the unusual diseases that has a separate common name for the fruit rot phase as for the foliar phase. So the fruit rot phase is called black rot. The foliar phase is called gummy stem blight. Very typical symptoms here on butternut squash. It's one of the fruits that is most susceptible. You can also get an internal browning on cucumber and muskmelon, and once in a while you'll get a little bit of black rot on watermelon. But for watermelon, which is the primary host I work on, is pygnidia that produce canidia that are exuded in the sticky matrix, so they are primarily dispersed by rain splash, and then pseudothecia that are produce ascospores, which we'll talk more about in a little bit. The other part of the disease cycle that I've worked on is the infested debris. And it's kind of surprising to me that a fungus that is so heavily dependent on moisture for disease development survives perfectly well in dry debris. Of course, the debris occasionally gets wet when it's out of the field, but the fungus also is very tolerant of these dry conditions. I've been involved in one major project looking at resistance, the potential sources of resistance to gummy stem blight, and this was working with H.M. Claus, where they had produced a number of crosses of watermelon Citrullus lanatus by citron, recently renamed Citrullus amarus, previously called Citrullus lanatus variety citroides. And so these pictures were taken about 10 days after Hurricane Matthew brought 15 inches of rain to Charleston. And so it's not very surprising that the susceptible Czech cultivars were about 98% necrotic foliage here on the left. But on the right, we had maybe 15 or 20% of only the older leaves that were diseased on some of the resistant crosses. It was really an extreme test of these resistant lines, and they performed very well. The problem was that none of the resistant lines flowered or set fruit because they were all sterile hybrids. And so the seed company, unfortunately, has dropped this project. I have heard, though, of two recent research groups claiming that they have found QTLs for resistance to gummy stem blight. I believe it's potentially in watermelon and potentially in muskmelon. And so I think there has been some progress made on resistance development, but there are no resistant cultivars for gummy stem blight in any cucurbit right now. So, of course, fungicides remain the major method of managing this disease. And the effectiveness does vary somewhat depending on the weather. The disease progress curves on the left there were in a wet season in the fall. The black circles on the left are the non-spray control, and you see that even with varying application frequencies of chlorothalonil, really about all we could do was delay disease progress, and that shift the disease progress curve over to the right. When we alternate chlorothalonil with systemic fungicides, and also the weather is more cooperative, we can get better disease control, significant reductions with chlorothalonil alone, or chlorothalonil alternated with sarcodinil plus fludeoxonil, which is switch on cucurbits, or Inspire Super, sarcodinil plus difenoconazole. And in this test, the fluopyram plus tebuconazole Luna experience offered significant improvement compared to chlorothalonil by itself. So on the left, we had significant yield losses, but on the right, there was no significant differences in yield among these treatments. So onto the taxonomy. So the old names for the fungus were Didymella browniae for the teleomorph and Phoma cucurbitacearum for the anamorph. In 2010, there was a major study of Phoma fungi with multilocus sequencing, and it just so happens that the Phoma cucurbitacearum isolate that was in that study grouped into a new clade, different from the other Phoma species, and the best name that was available in the literature for this clade was Stagonosporopsis, which is a genus erected in about 1910 or so. Now, of course, kept the same species name, and so the new taxon or nomenclatural synonym of Didymella browniae is Stagonosporopsis cucurbitacearum. Then Maren Brewer at the University of Georgia, when she started her program, I sent her a number of isolates from my collection, and she looked at them with the same multilocus sequencing and found actually there were two other clades within this group of organisms that did not match cucurbitacearum. One of them was a completely new lineage, and so she named that Stagonosporopsis citrulli from Citrullus watermelon, which is the most common host for for this species in the southeastern United States. And then she also found that some of the other isolates actually were identical with the species caricae, which used to be Phoma caricae, which comes from papaya. And when she tested the isolates of Stagonosporopsis caricae, they indeed infected both cucurbits and papaya, which to me is kind of a strange post-bridge between these two crops. I mean, obviously they're both grown in the tropics, like Central and South America, and perhaps that's where these fungi then originated, where both of these hosts were growing together. Some of the other changes, though, in nomenclature, of course, are because of the one fungus, one name provision of the International Code of Botanical Nomenclature, which means that we don't worry about anamorph and teleomorph names anymore. It's one name, Stagonosporopsis, 3 species. And certainly this provision makes communicating with non-plant pathologists much simpler in that we don't have to confuse them with two different names. And there are several other examples of changes in taxonomy and nomenclature in cucurbit pathogens. The Diaporthe, Melonis, Phomopsis cucurbitae group on cucurbits is all, actually there's now 4 species of Diaporthe on cucurbits, and I believe one of them is actually Diaporthe soji. So again, we have a cucurbit soybean pathogen connection. And then Myrothecium, which has been renamed Paramyrothecium. My student found some of these isolates on watermelon, and when he started to identify them, he actually found that they were members of 6 different genera. And that's a manuscript that he still has in preparation. So Maren Brewer and her postdoc also developed a simple multiplex PCR to distinguish these 3 species. And these are 3 primers used together, and these are based on microsatellite primers that she used. And so you see for Stagonosporopsis citrulli reacts with all 3 primers. There's faint bands here for Stagonosporopsis cucurbitacearum reacts with 2 primers, and caricae only reacts with 1 of the 3 primers. And so I consider this sort of to be a simple but very reliable way to distinguish the 3 species. There have been different names assigned in the literature to these 3 species over time. When I first looked at isolates in the 90s, I was able to distinguish them based on rapid amplified polymorphic DNA markers. And so we could distinguish the different species with rapids, and also distinguish them from Phoma species that are non-pathogenic. We isolated from cucurbits, but when we tried to re-inoculate cucurbits with them, they're either non-pathogenic or weakly pathogenic. I had a PhD student that then developed primers, and he also found that he had one isolate from New York, a caricae, that could also be distinguished with rapid primers. Another student found that they could be distinguished with AFLP primers, and then we ended up with the nomenclature changes based on multi-locus sequencing. There are 3 ATCC isolates of Divino albarani that are commonly used in many different studies, and actually all 3 of them from Europe and all 3 from cucumber turned out to be Stagonosporopsis citrulli. And so there have been some recent new reports, not necessarily of new hosts, but these are really more reports of where these species occur throughout the world. And so you see that all 3 species are reasonably well distributed around the globe. There are credible reports, some older reports based on morphology on a number of huge range of non-cucurbit species that are probably based on misidentifications. There is probably a credible report on a non-cucurbit water spinach, which is an Ipomoea genus in the sweet potato family, and that's based on beta-tubulin sequences of those isolates. But there's also a report on tobacco that I don't think is credible, again, because that was based on unverified ITS and beta-tubulin sequences. So as much as I would like Stagonosporopsis species to be pathogens of only cucurbits and to keep things simple, obviously, because some of them can also go to papaya, it's not improbable that they may also go to a few other hosts. So let's talk about spread of Stagonosporopsis species on land, and this is primarily by movement of seed. And one of the key papers here was from the Danish Seed Science Center in 1984, where they actually dissected seed and found that embryos are infected with the gummy stem blight fungus. So seed transmission, at least in cucurbits, is often assumed because, seedlings grown as transplants in the greenhouse come down with gummy stem blight. But, if you look at these reports critically, you have to recognize that there may be other sources of inoculum, such as carryover of crop debris in the greenhouse or airborne astrospores. So this is sometimes really only circumstantial evidence. I'm not saying it should be totally discounted, but in order to really prove that this is a seed-borne pathogen, you have to work with infected seed. And so there's one good, well-documented grow-out done in England from infected cucumber seed. I've worked a little bit with infected watermelon seed. There was the seed dissection study, and then also PCR primers that were developed at the University of Georgia, and I supplied the infected seed for that. Now that was based on fruit that I inoculated, and then seed turned out to be infected. So, for example, early collections from greenhouses could be an example of seed-borne inoculum. Here are two collections made in Ithaca, New York in 1891 when the fungus was referred to as Phylosticta cucurbitacearum, both from cucumber, one from the Cornell University greenhouse collected in January. So there probably were not viable astrospores floating around in Ithaca, New York in January. And I guess you would hope that in a university greenhouse they had done a good job of cleaning out the greenhouse. So I think we can eliminate other sources of inoculum in this case. So Dudley also collected gummy stem blight again on cucumbers in June of that year. That may be not quite such a clear-cut case, but some early reports suggesting seed-borne inoculum. Two of the isolates that I worked with from seedless watermelon seed that was supplied by the old pedo seed was seed produced in China. One of those isolates was part of Stewart's study, so that is definitely Stagonosporopsis citrulli, and one of the other isolates was part of two other studies where we were developing primers for these species, and also came out to be Stagonosporopsis citrulli. There probably are examples of the other two species that are also present on seed. It's just I don't know of any references to that have been published on those. Are there any quick questions at this point?
I have a question, Tom.
Yeah.
How does a leaf blight disease get called gummy stem?
Well, that is a very good question. I guess from the cankers, that I think used to be more common on cucurbits when they were grown without plastic. When I see cankers on cucurbits, I immediately think of soil-borne inoculum, that is from debris from a recent previous crop carrying over in the soil. And the gummy part is when there's damage to a cucurbit stem, sap begins to ooze out, and when it dries, it kind of looks like an amber-colored resin on the stem. Now that is not it occurs frequently with cankers of gummy stem blight, but it's not a diagnostic symptom, because you can sometimes see that with fusarium wilt, or you can also just see it from injury to the cucurbit stem.
Just taxonomy, are you more or less, is this just going to where we would have to go to multi-locus sequencing to really identify it? There's no other...
Well, you can use the primers that
Okay.
Aaron Brewer developed.
Oh.
The multiplex primers work very well. You don't need, yes, you don't need to go to multi-locus sequencing. Now, for other, organisms where people haven't developed the primers, yes, a lot, unfortunately, like, some of the alternaria or species on cucurbits, you have to go to multi-locus sequencing.
I wasn't sure if that was good enough to use for...
Yeah, I think, yes.
More or less.
Yes, her primers are, yes. Okay, so. Yes.
So, if I'm correct, if I remember correctly, cucurbits originated from North America, or like North Central America. And then soybeans originated in Asia. Do you have a hypothesis for how,
Well, actually,
or like when?
Well, actually, based on, we'll get to this later in the seminar, but the center of origin for the entire family of cucurbits is considered to be Asian, based on new taxonomy. And the paper shows how they potentially spread to different continents, but from Asia. So that's an interesting observation that both hosts for like the diporthy potentially were growing in proximity to each other. Most of this work was done by my recent PhD student, Gabriel Renberger, and it's still in preparation. As I mentioned, the infested watermelon debris is a major source of, I consider to be probably the most important source of inoculum for epidemics of gummy stem blight. And on this debris, you'll get both pycnidia and pseudothecia, although in the fall, I would say about 75% of these fruiting bodies would be pseudothecia producing ascospores. The fungus is bitunicate, so the ascospores are forcibly ejected at maturity. And so this was some of my early studies where I simply looked at survival of the fungus on cucurbit crop debris. In this case, I was culturing pieces of debris. And so for watermelon vines, which are really not that thick or that resistant of tissue, the fungus basically survives as long as the vine segments are intact, which in this year, the study was about 32 weeks after we placed them in the field. And these were naturally infected vines. Similarly, then working with naturally infected muskmelon crowns, as you see the large bleached area here at the crown of this muskmelon plant. So 1 year of the study, they survived for more than 68 weeks, since we didn't quite reach zero here, with crowns that were left on the top of plastic mulch. So that is on top of the raised beds covered with plastic mulch, which is the way practically every vegetable crop, except maybe squash and sweet potatoes, are grown in the southeastern United States. So there was a difference here, longer survival on soil surface or on top of plastic mulch, and more rapid decline of the fungus when the crop debris was buried. On the left, though, we did not see an effect of burial on that debris. But what I sort of, I guess, in hindsight, naively assumed was that this debris, the fungus was dormant in this debris when it was sitting on, after the crop was over. But what my graduate student came up with this simple little assay where he took these naturally infected pieces of watermelon vines, cut them up, stuck them to the top of petri dishes with Vaseline, and then allowed you to see some younger colonies of ascospores, maybe three different ascospores were ejected here, and then the more typical look of the fungus when it's grown up: olive green mycelium, sort of substrate mycelium, sort of whitish aerial mycelium, and then fruiting bodies scattered out in there. So using this assay then, he monitored release of ascospores from a field, in both cases it was a field that I had used for a fungicide trial. So pretty much 100% of the lines were disease at the end of the season when he started sampling, collected vines, monitored percentage of vine pieces that released ascospores, and basically went until about, started sampling, I think in 2018 was December, and in 2017 was January, and basically went to about July in both years. And when there were none or very few pieces that were still producing ascospores. So a slightly different shape of each curve because he started out with a higher percentage in 2018 than in 2017, but in terms of management, it's more important, I think, that they both ended up at about the same point in time. We also monitored ascospore release in in one different way, and that was with trap plants, where we put out plants of watermelon and cucumber, 3 of each host at various distances from the field, expose them for 48 hours, and then put them into a moist chamber for development of gummy stem blight. He also tried to use this volumetric spore sampler to collect airborne ascospores. Was never able to detect any ascospores in his samples. And he was using a PCR reaction to try to do that. We had borrowed the spore sampler from Katie Stevenson at the University of Georgia, who had tried the same thing about 10 years ago, and she was then trying to look for spores under the microscope, and also could never find any ascospores in her samples. So neither of us is quite sure why this technique doesn't work for Stagonosporopsis. It has worked for other foliar pathogens for trapping spores, but neither of us were able to detect them that way. So here are some typical symptoms that we saw on trap plants. This water soaking here on the stem of watermelon, or 3 lesions here on this leaf of cucumber. And so this is the relationship between incidence of plants and distance from the spore source for 2017. So that was sampling from October again through June. So we basically, once he stopped detecting ascospores in the plate assay, he stopped putting out trap plants. And so there's a good relationship here on decreasing incidence with increasing distance. And there was no difference in this year between watermelon and cucumber. But in 2018, we had actually higher detection with cucumber than with watermelon. And we also detected ascospores at a farther distance from the field. 366 meters, I think, was the 1,200 feet distance from the spore source. So not a huge distance, but clearly airborne ascospores can be an important sort of short distance dispersal mechanism for this pathogen. Then we also did this experiment under controlled environment conditions, and that was we put debris that we had taken from the field in the head house of the USDA building in which the Cultural Research and Education Center is located. Half the head house is about 190 or 200 feet long, so we could space plants up to 180 feet away from the debris. We set up a drip irrigation line to wet the plants because, like I said, we're not going to get infection if the plants don't have some leaf wetness. And we also set up a couple fans to try and move the debris, move the spores from the debris throughout the head house. And we did this 3 times, 2 times with debris, and then a control experiment without debris, when fortunately we didn't detect any infected plants in that experiment. And we didn't detect disease on both hosts in both experiments. Obviously you see we had 1 or 2 plants that were infected at I think it was 120 feet in experiment 1. Not as much spread in experiment 2, but at least a short distance spread that I think confirms that, the debris was very likely the source of inoculum for these plants. So, before we move into the last part of our topic, any questions at this point? Okay, spread of Stagonosporopsis by sea is a little bit more theoretical or speculative. But the idea comes from this paper with the short title, Gourds Afloat. This is the paper that proposed the Asian origin of the Cucurbitaceae based on multi-locus sequencing and phylogenetics. So this is a list of 22 genera in which there are one or more hosts that are susceptible to gummy stem blight, all different genera of cucurbits, and the three that I'll focus on that the paper specifically talks about are Lagenaria, which is bottle gourd, Loofah, which is loofah or sponge gourd, and Sikios, which is bird cucumber. And so in their analysis, molecular clock analysis based on the rate of mutation and the number of mutations they found, they said that loofah seems to have evolved about 35 million years ago and also spread from Africa to South America, presumably by the dried loofah fruit floating across the South Atlantic Ocean. Similarly, Sikios evolved about 8 million years ago and is thought to have spread from South America to Hawaii, through the Pacific Ocean, and then possibly on to Australia. Lagenaria, about the same time, 8 million years ago, and again spread from Africa to South America. And then just for perspective, two of the edible cucurbits are more recent in the evolutionary history. It's 3 million years for cucumber, center of origin Asia, and 2 million years for watermelon, with the center of origin being Africa. So the hosts were clearly in place before the pathogen can be detected in a molecular time clock analysis. Because the first species, Stagonosporopsis caricae, presumably separated, evolved from the ancestral species only about 73 million years ago. And then from what was left of the ancestral species, Citrulli and Cucurbitacearum diverged about 11,000 years ago. Now, Brewer and Stewart suggested that this differentiation and specialization on cucurbits was possibly influenced by the domestication of cucurbits and the movement of especially squashes from South America into North America. So the idea was that there was some type of advantage for these organisms to specialize on cucurbits as opposed to sticking with the multi-host lifestyle of Stagonosporopsis caricae. So this is sort of a graphic of that. Here you see Stagonosporopsis caricae as the ancestral species and Citrulli and Cucurbitacearum as sister species that evolved from that. And you can see the same pattern if you just look at the IS2 sequences, where my student found that Stagonosporopsis caricae, there's only one base pair change in the IS2 sequence. That's why in many cases for Ascomycetes we need to go to multi-locus sequencing. ITS is not specific enough. So these are symptoms on loofah. They're in the left leaf spots. Notice loofah is one of the more resistant or less susceptible cucurbits than certainly watermelon or muskmelon is. The leaf spots never get quite as large on loofah. We did not see any fruit infection though in this study on loofah. This is on Lagenaria or bottle gourd. Again, it's also one of the less susceptible cucurbits to the foliar blight, but the fruit are very susceptible. Now, I cannot honestly stand up here and tell you that all of this disease is caused by Stagonosporopsis because the fruit are also very susceptible to Keinath. And it's difficult to distinguish those two symptoms of those two fungi, or just distinguish those two fungi based on symptoms. But I do think because of the extensive water soaking on this particular fruit, there is some Stagonosporopsis on this fruit. And so, to sort of complete the story about movement by seed, I found this paper by Thomas Whitaker, who was one of the 20th century cucurbit botanists who looked at oceanic drift of gourds. And so he floated gourds in an enclosure in the Pacific Ocean out in California and left them there for up to 224 days and then germinated seeds from those gourds and found that really floating the gourds in seawater did not really reduce viability of the seeds that were in those fruits. And then to take it one step further, he also directly immersed seed in seawater. And again, looked at germination and found very, really not much of a decline in germination of the seed after they were immersed in seawater for up to 96 days. So by extension, if Stagonosporopsis was in seed of these gourds that floated to South America, since the seed were still viable, my assumption is that the fungus is probably also still viable in those seed. And so could have traveled with the gourds from Asia, potentially to Africa, and then potentially to South America. So seed is certainly a refuge, a survival structure for both the host and the pathogen. And I think there are many advantages that seed-borne pathogens have. Probably the most important one is that they are automatically dispersed in a protected environment when the seeds of the host are dispersed. And being present in the seed allows them to take a lot of shortcuts in the disease cycle. They don't need to produce spores or to have their spores dispersed. And they also don't need to wait for a conducive environment for spore production or spore dispersal or even spore germination because they are already there, ready to germinate when the host germinates. Now, one other thing that Gary and I talked about, and it's an idea that came from Dr. Jurgensen at the University of Copenhagen, is that these seed-borne pathogens go into a hemibiotropic phase when they infect the seed because normally Didinella is a very aggressive necrotropic pathogen. It's trying to kill the host, but its actions in dissolving the host, pectin and cellulose end up killing the host. But it doesn't, when it's seed-borne, it's not doing that. The seed remains viable. It's not killing the seed. And so I don't know if the fungus is simply going dormant or if it's really entering some other temporary type of life cycle, which would be considered hemibiotropic in that it's sort of biding its time, not attacking the host for a certain period of time until whatever happens after germination that triggers it to resume its necrotropic phase of its life cycle. So to summarize this presentation, we're now dealing with 3 morphologically identical but genetically distinct species of Stagonosporopsis and cucurbits. Citrulli has certainly been isolated from seed, and I'm sure that someone has isolated Keinath and Cucurbitaceae as well. Seed has been an important dispersal for this fungus, allowing it to have a worldwide distribution. Certainly by land and potentially also by sea. And airborne ascospores are yet another way of dispersal of Stagonosporopsis citrulli, which is what we used in our studies, but probably also Keinath and Cucurbitaceae as well. I'd just like to acknowledge my lab group, Casey Conrad on the left, temporary grant technician, Sean Toporek, a master's student en route to PhD, My younger daughter Caroline, who worked in our lab last summer. My PhD student Gabriel Renberger in Orange. He is currently a postdoc at the USDA lab in Fort Pierce, Florida. I'm working with Scott Adkins and Bill Turacek on area-wide management of whitefly transmitting viruses in cucurbits, and my lab manager, Julie DeVos. Much of my recent work on watermelons and gummy stem blight has been funded by the South Carolina Specialty Crop Block Grant Program. So with that, I'd be happy to take any other questions you have.
So residue is like the most important source of inoculum. So what do growers do with their residue?
Well, I have, since I've started sort of on this theme a number of years ago, I have gotten them, pretty much all the watermelon growers in South Carolina, on a 2-year rotation cycle. So certainly either 1 or 2 years out of cucurbits, which is long enough for most of the debris to decay. And I think they have seen the benefits of that, since some of the very large growers are doing it. Also, another reason for rotation is also their attempts to control fusarium wilt, which of course don't really have much. That short of rotation doesn't have any effect on fusarium, but they are seeing benefits on control of gummy stem blight. And then, a fairly rigorous spray program starting at flowering, because as I tell them, the first spray of the season is the most important spray because it will help to delay disease initiation. But pretty much once you start spraying, in a normal season, they have to be on pretty much a weekly spray schedule all the way through harvest. So it's mainly crop rotation and fungicides. Yes.
So if you have 3 morphologically identical fungi, but there could be 3 different species, from a diagnostic standpoint, if a clinic receives a sample that's from the stem blight, is there an advantage to identifying the fungus to species as far as control recommendations?
That's a very good question. In this case, yes, because resistance to DMI fungicides has been found in Stagonosporopsis caricae, but not in citrulli or cucurbitaceae. And people have looked. I mean, I've looked, and they've also looked at the University of Georgia, and isolates, the DMIs, especially tebuconazole, has been used in watermelons certainly for the past 15 years, and we have not yet found. And it is a very popular fungicide because the generic form is very cheap. And so it is widely used, but we have not seen resistance yet in citrulli, but some of the isolates of caricae are resistant. So from that perspective, for making fungicide recommendations, especially since it's a widely used fungicide, that is something that you would want to caution growers against not using if you have resistance. So you would either have to check isolates for resistance, or you would have to, it would be quicker to simply identify them.
Something earlier about host resistance. Is there any breeding work going on that? Or
yes and no. There's a lot of work. There's work going on to identify sources of resistance. But we really don't. I mean, there have been 3 or 4 other studies, at least that I know, because I supplied isolates to the people who were doing the screening. But for whatever reason, the resistant lines that they found have not progressed to the release of resistant cultivar. So I don't really know how much active breeding is going on at the moment, but certainly people are certainly looking for good sources of resistance.
Yeah, I saw on Twitter today, I think, that the EU had banned chlorothalonil, and it looks like you did pick on it.
Well, I do. I've been a very strong proponent of chlorothalonil for control of gummy stem blight, because I think it weathers better during rain than mancozeb, which is the other commonly used protectant on cucurbits. Some of the growers used to be a little, and they still are, the reason they use mancozeb more often than chlorothalonil is because the chlorothalonil label has a caution about potential phytotoxicity in fruit under hot, sunny conditions when it's sprayed. Now, in the study that I did back in the late 90s, one year we had more fruit injury with chlorothalonil, and the other year we had more fruit injury with mancozeb. And I have not really been able to reproduce that phytotoxicity. So more growers are willing to use chlorothalonil up to, certainly they use it in the early part of the season, and there are a few growers who will use it through the entire season. Certainly in the fall, we don't have problems with, for the fall crop, we don't have problems with with sunburn or phytotoxicity. I do think some of what was called chlorothalonil phytotoxicity was actually sunburn of the fruit. And it just happened after they had just sprayed chlorothalonil. But yes, that is somewhat of a concern to me, yes.
You mentioned a few other fungicides in some of the data that you had, but it seems to me like they were better. I can't remember the combinations, but it seems like they were better than chlorothalonil, they're more expensive.
They're more expensive. And that, growers in South Carolina do not like to spend a lot of money on fungicides, even though, I mean, for powdery mildew and anthracnose, you can make very strong cases that you will get a tremendous yield increase by using fungicides on watermelon to manage those two diseases. Unfortunately, I cannot make quite as strong of a case with gummy stem blight, even though I've been trying to do this for 25 years. And I don't know if that's because fungicides really don't work that well, even the best fungicides don't work that well, or if watermelon is just more tolerant of the damage caused by gummy stem blight than it is, for example, the damage caused by powdery mildew. I have not been able to get good statistical evidence that spraying all these fungicides for gummy stem blight is automatically going to increase your yield. I mean, it really looks like it should, but another part of the problem is that gummy stem blight develops so quickly. You can have it come in like in the last 3 weeks of the crop cycle. You'll end up with 75% defoliation at the end of the season. Your plots look awful, but the yield has already been set. And you may get a slightly smaller melon out of it, but you're not going to get fewer melons. And it also does not seem to have as much of an effect on sugar content as cauliflower does. So, from the foliar symptoms, it looks awful, and the growers really are quite, concerned about this disease. But I don't really have the hard data to say that this is a serious yield loss disease in watermelon.
Any questions? Okay, thanks, Tony.